I'm Dr. Nazer Kwasi. I'm the biology lab manager at the WAVE program. Here, you are in the biology lab where we conduct a lot of research. All the samples collected from the survey are brought here in this laboratory. And here we can do DNA or RNA extractions. And then after we got the RNA or DNA, we start the detection using different methods. The first method that we use here is the PCR method for detection. We amplify the DNA. And also the second one that we use here is reverse transcriptome PCR for detection of RNA, because we do the cDNA. And the third method that we use here is the quantitative PCR, which is real-time PCR for the amplification also for DNA. And also we can do the quantification RT-PCR also. After we have some DNA amplified, we can clone directly the DNA that we got, the fragment, the amplified fragment. We can clone them in a vector and then send them for sequencing. We can also do, and we do amplification using RCA method, which is a rolling cycle amplification, and so on. We have a lot of techniques that we can do here.
I'm Kwasi Modeste, the manager of the Plant Tissue Culture Lab. And in this lab we work mainly on sanitation of the plants using medicine culture plus thermotherapy. And after sanitation we do the conservation. And here, for example, we work on cassava. And we are trying to conserve these adopted cassava varieties. So we have 22 adopted cassava varieties. We are also working on sweet potato, and in conservation we have 16 elite varieties of cassava. So after sanitation, conservation, we are also trying to do mass propagation in order to provide planting material to the farmers. So just to give you an example of our capacity, this is the type of container we use because it's very cheap. We purchase this type of container on the local market, very, very cheap. So, but we have, and in this container we can put from 7 to 10 plantlets, explant that will develop into plantlets. But so we have this possibility to reduce the height of the container, but to conserve, to keep the number of plants. Here we have 5 growth room, and all this growth room can contain 9,000 of this type of container. So if you do the calculation, at once we can have 90,000 of plants. So we have this big capacity, this ability to provide planting material to the farmers. When we do the mass propagation, we also, outside of the lab, we have some facilities to do acclimatation. in forms of tunnels of acclimatation, greenhouse of acclimatation. So from this, after acclimatation, we can directly yield, provide this planting material to the farmers.
I'm Dr. Linda Leabo, lab manager at WAVE HQ in Côte d'Ivoire. So I'm managing the laboratory of valorization of symbiotic microorganisms, and this laboratory aims at working on the diversity of beneficial microorganisms of a soil. Specifically, we will work on the symbiotic fungi, the arbuscular one, the valorization of symbiotic microorganism, VSM. In this lab, we are assessing the diversity of arbuscular fungi countrywide for identification of the best one that can be used in field experimentation in the scope of improving, boosting the performance of cassava, yam, sweet potatoes, and other food crops in Côte d'Ivoire. Taking advantage of epidemiological assessment of cassava mosaic disease in Côte d'Ivoire, we collected soil sample from fields prospected in 2020 according to the harmonized protocol of WAVE. Composite soil were sampled from each field and brought in the lab here at WAVE HQ at VSM lab and stored at 4 degrees. After 1 month, soil spores are extracted from this soil in order to be identify the species, but also to run trap culture for further analysis. VSM lab opened in June 2020, and we started with the soil collection in September. So actually we are really at the beginning of our activity, and the next step would be starting the trap culture for identification of best species for field experimentation.